Welcome back to Dr. Liu's VCE Biology. Today's topic is changes in species over time, as presented in the 2018 VCE Biology exam. Let's learn together. If teachers, parents, or students notice mistakes in my explanations, please provide me with your feedback. The photograph shows a fossil of the ornithopod dinosaur Diluvicosa picaringi found in 113-million-year-old rock in Western Victoria. The fossil consists of a tail, a partial hind limb, and some vertebrae. D. picaringi grew to 2.3 m long. The diagram on the right is a reconstruction of the complete dinosaur. Evidence suggests that the dinosaur was fossilized in a log-filled hollow at the bottom of an ancient riverbed. Two stratigraphically younger fossils that had been found previously at a nearby site are closely related to D. picaringi. Two closely related fossils, found previously at a nearby site, are stratigraphically younger than D. picaringi. Because these related fossils are younger, they were recovered from a shallower depth closer to the present-day ground surface than the rock matrix containing D. picaringi. The close common ancestry shared by Victorian ornithopods and related fossils from Antarctica, South America, and Africa is attributed by paleontologists to the fact that these continents were once connected to Australia as part of the supercontinent Gondwana. P. 3500 Ma represents the emergence of the first primitive prokaryotic cells. This event occurred before cyanobacteria, which themselves are a type of prokaryote. Q. 800 Ma marks the critical appearance of eukaryotic cells. This leap occurred between cyanobacteria and corals, complex animals, as eukaryotic cells were a necessary precursor to all subsequent complex multicellular life forms. R. 400 Ma signifies the successful colonization of land by major groups, such as amphibians and insects. This diversification occurred between fish and mammals, at a time when the land masses of Gondwana were still largely connected. Natural variation in the phenotypes of the allele sagray lizards across the islands of the Bahamas is essential for evolution and arises from several genetic mechanisms. This variation exists primarily due to mutations, which are the fundamental source of new alleles not previously present in the gene pool, and through sexual reproduction, which constantly shuffles existing genetic material via processes like recombination and random fertilization to create unique allele combinations. Less frequently, but also contributing, are larger changes in the genetic code, such as those involving chromosome number, which can dramatically alter an individual's phenotype. In 2004, a hurricane killed all populations of A. sagray lizards on 7 of the smaller islands. Scientists randomly chose 7 males and 7 females from the remaining population on a large island. They introduced 1 male and 1 female to each of the 7 smaller islands. Over the next 3 years, the scientists noted that the size of the populations increased on each of the 7 smaller islands. The scientists measured the genetic diversity within each of the populations and found that there was lower genetic diversity in each new populations compared with the population on the large island. The lower genetic diversity in the new A. sagray populations is a direct consequence of the founder effect. The hurricane caused a severe population bottleneck, and the subsequent establishment of new colonies by just 2 individuals meant that the founders carried only a small fraction of the original population's alleles. Consequently, any alleles absent in these 2 individuals were permanently lost from the new gene pool, resulting in a significantly reduced variation in the descendants' phenotypes. The rapid decrease in the average hind leg length of the lizards on the smaller islands was due to natural selection driven by different selection pressures in the new environment. Over this 3-year period, those lizards with shorter hind legs had a selective advantage, making them more likely to survive, reproduce, and pass the heritable short-leg trait to their offspring. This differential survival explains the change. The student's incorrect assertions that the change was due to the lack of use of the hind legs or a need driving the change are contrary to modern evolutionary theory, which confirms that change is caused by the environment acting on pre-existing genetic variation, not by effort or desire. Deep time and geological context. Q26, Gondwana, the ancient supercontinent where similar fossils across continents originated, providing key biogeographical evidence. Q25, fossils. Adheres to stratigraphy, younger fossils are found in shallower depths. Q27, timeline. Life began with prokaryotes, p. 3500 ma. The emergence of the eukaryotic cell, q. 800 ma, enabled complex life, followed by the colonization of land by groups like insects and amphibians, r. 400 ma. Lizard case study, genetics and selection. Q7a, a variation. Phenotype differences are fueled by mutations, new alleles, and sexual reproduction, a recombining existing alleles, providing the basis for natural selection. Q7b, a founder effect. Establishing new colonies with only two individuals caused a founder effect, leading to a significant reduction in genetic diversity, allele loss. Q7c, selection. Different selection pressures on the smaller islands favored lizards with shorter hind legs. This differential survival and reproduction caused the population's average leg length to rapidly decrease. If you need my help, please add me on WeChat or WhatsApp and send me an email. Thanks for your attention. Please note Dr. Liu's VCE Physics channel, Dr. Liu's VCE Chemistry channel, and Dr. Liu's VCE Biology channel.